Today we have a presentation by Brandon McNellis. McNellis is a research ecologist with the U.S. Forest Service at the Institute of Pacific Islands Forestry. His presentation is titled Plant Demography Provides Key Insight into the Structure and Function of Hawaiian Forests. His research is conducted through a HETF permit. Please note that permits are required when entering the HETF for research purposes. And Brandon, you may start whenever you're ready. Mahalo.
Thanks a lot, Tristan. Okay, I do not use Zoom that much, so apologies if I don't get it quite right. Can everyone see that okay? Does that work, Tristan?
Yep, looks good. Mahalo.
Perfect. Okay, I want to start with saying that I just came down with some sort of upper respiratory infection yesterday, so I'm gonna do my best, but apologies if that gets in the way during the presentation. But with that being said, today I would like to talk about plant demography and how it can be used to provide insight into the future of Hawaiian forests. I'm going to talk about two projects that I'm working on the Institute of Pacific Islands Forestry. And there we go, next slide. So this is a longer presentation. And so I have it broken down here into three parts. I'm going to start off by talking about what is offered by studies of forest demographics, some examples of demographic studies, questions that can be used to track plants through time. Two questions I am asking these two projects is how do forest demographics in Hawaii compare to other forests, as well as how will forest composition change novel fungal pathogen death or rot. The first question is going to be the second part. Demographics in Hawaii compare forests. I'm going to do that using the neon. I'm going to talk about the site of Pu'umaka'ala, Hawaii's only neon sites, what it looks like, and about how the local ecological Hawaii is really important and critical for understanding the ecology of place. And towards that, I think a site narrative will help improve the inference data collected at Pu'umaka'ala. And I'll finish by talking about Pu'umaka'ala and how it compares the two network. The second will be about third part of the presentation, and it will be how are these forests going to change due to this using two forest geosites. They're in Hawaii. One of them is critical to experimental forest infrastructure. And that's a Lapahoi. It's a mature ʻŌhiʻa. Also has rod. Acacia co-occurs with ʻŌhiʻa, and we're going to use individual-based models to predict the consequences of this mortality. And I'll talk about what like 75% dieback looks like, is what questions we can ask with an IBM. So the first thing, what do demographics give us? Plants can be tracked, studies by cover, stand, or level using a variety of sensors. On the left, an older study that looked at the recovery of ʻŌhiʻa on fresh lava, and lava flows of particular years on a basal there on the left. And the question this was answering was, how long does it take ʻŌhiʻa to really recover its biomass after it gets into day? They found 300-400 years it starts getting going. On the right is a similar sort of study looking at vegetation on abandoned sugar canes on, I believe, the Hamakua. And this is on a percent basis, and it's showing here that years post abandonment, using remote sensing data about most of the area covered to pre-disturbance levels to reference conditions. individual based models are really a way to go with these things, because then you control trees and how they affect and how they control mortality and stem turnover within a forest. Studies are both from the plots. The left is from Faith Inman 14, looking at the relationship growth rates and on 4 species of trees in Laboi, the differences, vital rates, and how they might affect the that's my window. Sorry. They might relate to other demographic processes. A study that looked at plant traits, how plant traits such as mass base leaves or Venetian architecture might and explain relative growth rates, graphic rates across this forest. So the first question I would like to ask is, forest demographics in Hawaii compared to down a little bit on the left is a paper Rebecca Ostertag and looked at the Lapua plots, and relative to other tropical forest plots here on the total trees per hectare are relatively similar to other tropical forests. YAS here, YAS is Yasuni, that's in the Brazilian Amazon. PL here, Palanan, that's in the island of Luzon in the Philippines, and we're similar to other tropical forests across the world, but we're not similar in terms of species richness. We're much more depauperate as far as the number of species that occur in our forests, and so I think there's still a lot more to be learned from how our forest compared to other forests across the globe. The second question is, how will forest composition change due to rapid ʻŌhiʻa death? This is a super recent paper from Vaughan 2023, looking at the susceptibility of current forest in 2020 to rod infection, as well as the predicted severity of rod across those pixels. And it stands to cause some major changes to forest demographics and cover across the island. So towards question 1, what can Hawaii offer to multi-site studies of forest demographics? I'm going to use the NEON data up here in the upper right. Use NEON data at Pumakala and compare our forests in Hawaii with other forests across the NEON network. NEON, if you're not familiar with it, has a bunch of sites across the United States, both in the lower 48 as well as Alaska, Puerto Rico. And we just have one little plot here in Hawaii. It is the only NEON plots in Hawaii, and it's actually the only one in the Pacific, in the tropical Pacific. So what does Puʻumaʻa look like? Well, it's your very typical ʻŌhiʻa overstory and tree fern understory forests. Although there are some areas that have a koa overstory and then this grass understory down here. It's a very diverse plot. Here it is in map form. It's up the Stainback Highway. The Kulani Correctional Facility is right smack in the middle of it. And it's got all of these little white dots or plots that are scattered around the natural area reserve, with a flex tower, this yellow diamond right here in the center. The thing that really separates our plots from the other plots of the NEON network is just how new they are in terms of geologically. Flow ages across the natural area reserve are as young as 1942 up here at the very top, and they can be as old as 11 to 30,000 years old, which is still super new in geologic time. No other NEON site really has soils that are quite that new. The second thing is precipitation. Mean precipitation at the tower is around 250 centimeters right here, but it drops down to as low as 200 centimeters. Up top, higher up the slope, and as high as 600 centimeters at some of these plots downslope, which relative to other plots in the network is absolutely nuts. The other new thing is disturbance by ungulates. I do not believe anywhere else in Neon has pigs. One site in Texas might have peccaries, I think they are, but they don't cause the same kind of ecosystem disturbance that pigs at Pumakala does. There are a lot of efforts to mitigate that. Lots of fences have gone in already, and pigs have been removed, but it's a pretty constant battle, and they're still in some areas turning over habu, eating out the pith, and just generally causing a mess. And you don't see any of this in the actual Neon data. This is what Neon data actually looks like when you open it up. It's just piles of numbers and spreadsheets. And that doesn't really represent what Hawaii is. There's a lot more nuance and a lot more complexity to it that I don't think comes through if you're just a scientist from the mainland that is looking at the data as part of a cross-site comparison. And I think this is an important distinction because local ecology is really important to a place-based understanding of an ecosystem. And this isn't a new thing. There's been lots of research that goes into place-based analysis and the idea behind places being important for understanding the ecology of an area. Especially in Hawaii, these bottom two references are from Hawaii directly. And I think that here, especially, it's very important to understand a lot of the nuance that goes into how that data is collected and where it's collected in order to understand it and apply it appropriately. So how do we do that? Well, I think that's an easy way to start would be to publish a site narrative, do an intensive comparison with Puamakala as well as other sites in the rest of the Neon network, and make some easy predictions. I think that, interestingly enough, Puamakala would be most similar climatically and ecologically to sites in the Pacific Northwest than to other tropical sites. I think that a naive view may be to compare Puamakala to other sites like Guanica, which is in Puerto Rico. But I don't think that's necessarily an appropriate comparison. And I think that being more explicit about that would be a really good contribution. Soil age and disturbance history, I think, will be really significantly related to ecosystem dynamics across PUM. I think that'll come out in the data. Here's an example of a cross-site comparison between Puamakala, which is here in Hawaii, Abby, which is in the Pacific Northwest, it's in Washington State, and then Guan, which is Guanica, Puerto Rico on the dry. You can think of it like the Kona side of Puerto Rico. You can really see the tropical character of both Guanica and Puamakala in their temperatures on the right, these black lines. And there's not a whole lot of movement seasonally with respect to temperature. A little bit more in Guanica because it's dry, it gets pretty hot. What's interesting is the precipitation pattern really follows the Pacific Northwest more than anything. These wet winters, dry summers, and even the magnitude is similar between the two of them. Wind as well. It's surprising that Puamakala doesn't get more wind, considering there are major disturbances from hurricanes, but they just don't pop out in the climate record. And I don't think they're as important to ecosystem dynamics as you would expect from the places that get hurricanes, such as Guanica. There's interesting patterns that come out in the understory vegetation. These are non-metric ordinations of the understory plant communities at all three of the Neon sites. Again, PUM is Puamakala, Guan is Guanica, Abby is in the Pacific Northwest in Washington State. Each black dot here represents a plot. Each gray dot here represents a particular species. And we're ordinating in plot and species space. There are 5 black dots in PUM for each plot. Each plot is really a plot year. And so we can group the years and see the trajectory of the ecosystem moving through time. There's not a very strong pattern in PUM. There's a little bit of one, but there's not very much data. And so I think in a couple of years, we'll know if there's some kind of succession going on in Puamakala. The really striking thing is the spatial relationship between the plots. Each one of these polygons represents a plot throughout all years, and plots are really separating in species space very distinctly. If you contrast that to Guanica and Abby, there's much less spatial organization of the plots across Guanica and almost none across Abbey. I think that a lot of that has to do with the relationship between plant cover and soil age, as well as disturbance history across Puʻu. There's a little bit of a time signature in Guanica, but it's very chaotic, probably because of high inter-annual variability in precipitation. It's a very dry site. And a little bit more directionality in Abbey, probably because of understory succession. So the next steps for this are to incorporate more data. I have structure analyzed. I didn't get into this presentation in time, but how does structure change across space and time at Puʻu, Abbey, and Guanica? Could other site comparisons maybe provide a better context to Puʻu? And then what other neon measurements are really special in Hawaii and need some sort of elaboration? For example, carabid beetle abundance here on the right. There's not super strong patterns here that I can see, but there was a statistically significant difference between years as well as between plots in terms of beetle abundance across space and time in this data set. The interesting thing is apparently carabid beetles here can be arboreal, which is different than other. I'm not a carabid beetle person. I have a different collaborator that's going to help me out on that part of the project, but specific parts of carabid beetle natural history may be unique to Hawaii and would require some special interpretation with respect to the standardized measurements across neon. And finally, can we generalize this approach across the entire neon network and really bring some of these Hawaii specific understandings to the whole data set? So the second project that I've started here has to do more closely with the Hawaiian Experimental Tropical Forest at Lapoi, and I'm asking, what are the demographic outcomes of a novel forest pathogen in Hawaii forests? This one was really important. This research was a workshop done with the Smithsonian Tropical Research Institute, and my collaborators thereof, Jessica Needham and Sean McMahon. So we have two sites in the Forest Geo Network, and the Forest Geo Network is a cool network because it's across the whole world, not just the tropics. It used to be just tropics in equatorial Africa, South America, and Asia, but now it includes sites in North America. We have several in Europe, one all the way down here in Victoria, Australia, and we got two over here. They're all ours. The Lapoi one is part of the Hawaiʻi Experimental Forest. This is a paper by Rebecca Ottertag from a few years ago that sort of outlined the structure of these plots, their location, and introduced them to literature. Here's a picture of Jonah. Jonah's our tech. He is by far the most important person for getting this data figured out and ready for me. So thanks, Jonah. And the Lapoi plot is on the wet side. It's near Lapoi. It's got these really big ʻŌhiʻas, super tall, very large, and it is a mature. It's one of the last bit of mature montane wet forests. It also has rod. It's just starting to come through, and those of you who don't know, rod is a relatively new fungal pathogen to the island. I feel like it got somebody who knows better than me, but like 2012 or something like that, it came through. The affected area has doubled from 7,000 to 121,000 hectares from 2016 to 2019, and so it's coming in very fast. And I think that Forest Geo data can very much inform the science and management on this dieback. Hopefully, it won't actually. We're taking steps to mitigate rod in the actual Lapoi plot. And so hopefully it won't actually come through, because there's lots of really good mature ʻŌhiʻa here. Here on the left, this is from the Lapoi plot in 2018. You can see the DBH distribution of the ʻŌhiʻa. There's lots of really big trees there, 100 to 150 centimeters DBH. Some really tall ones, 15, 20, 25 meters tall. This is allometricite. But we also have a lot of acacia, a lot of very big acacia, 50 all the way up to 110, 120 centimeters DBH. and very tall. And so it's co-dominant in some places the Metrosideros. Besides the Sibodium, Sib, and the Chirodendron here, Metrosideros is definitely the most abundant stem on the plot. Although it's not growing super fast, it's relatively slow growing, and you can see acacia, despite being not very abundant here, is growing very fast. It's definitely in the plots. It's coming in, and I wondered what will happen if the Metrosideros dies back from rod. What will happen with the acacia that can come through? I think a way to answer that, if we're not going to let the forest actually die back, is with an individual-based simulation model. IVMs can be used to model the survival, growth, and reproduction of all of these individual stems through time. They can also be used to model the recruitment of new individuals into the data set and then death from competition. And it can do this over and over again for each census cycle and predict how the forest will change through time. The really cool part about this is after we fit this workflow to our data, we can then run experiments, but like introducing a novel pathogen to the simulation and design experiments to look at the potential outcomes on forest composition of these sorts of events. So for example, here on the left is all of the Metrosideros and acacia that were alive in 2018 on the plots. Black is Metrosideros, and blue is acacia koa. Now on the right, and you can see here on the left still, it's super like heterogeneous populations, really big densities down here in the bottom right. The underlayment is elevation, so you can see we're a little bit higher elevation down here in the bottom left, and then it goes down slope up here to the top right. Big patches here to the left on the upper ridge of Metrosideros, and then down here to the right. And koa generally spread all over. We can simulate a dieback event, like let's say 75%. And here I've just taken 75% of the trees and killed them in simulation space. And there's still a lot of Metrosideros left, but now there's tons of space for the koa to come through. We relieved a lot of competitive pressure on the koa. And they have a lot more room, a lot more light and a lot more room to compete with the Metrosideros for dominance or co-dominance of the canopy. And so what'll be the effect of this if there really is 75% dieback of the Metrosideros? How will koa in particular? Well, we can run this with any species that are on the plots. How will those populations respond to the dieback? A big question that I definitely need to talk to some rod collaborators on is dieback, is rod death size dependent? And if so, how does this affect recovery? From these two graphs, you can see that Metrosideros has a pretty stable population. There's all these big mature individuals and then a ton of seedlings down here in the understory. And so does rod only affect big mature individuals? And if so, are these small individuals going to be able to then be released from conspecific competition and fill out the overstory? Alternatively, the acacia is doing quite well. It has a really big cohort right here at about 40 centimeters. Those guys will be released from competition too. And so how are these demographics going to change over time? Importantly, are there mortality thresholds where ʻŌhiʻa just never recovers? Like, can it come back from 95% mortality? Or are there points where it will simply be outcompeted by either acacia or other understory natives or even invasives? We can model those also. So those are my two projects. With that, I will say thanks to all my co-PIs and collaborators and technicians and organizations, and I can take questions.
Mahalo, Brandon. I don't see any questions in the chat currently, but if anybody does want to jump in and ask something, you can feel free to now. If no one else has a question, I guess, Brandon, I have a question for you. Have you modeled the impacts of non-native species and how they would change over time? Or is that not something that you've looked at yet?
I haven't looked at it yet. There was some work done on invasives when the plot was first established. Becky Ostertag did some of that in their initial characterization of the plot. It's something we could model if we had demographic information on the invasives as well, but I haven't seen that data yet.
That was my question as well. This is Julie Dinslow. Thinking about the impact of strawberry guava on canopy opening in these plots. From our demographic data on strawberry guava from other plots sort of around the corner, it looks like strawberry guava would be released.
Yeah, I think that it probably would. I think the strawberry guava in particular would be hard to model in this situation because it totally dominates when it does get released. It forms those super thick stands. And I don't know if a model would be able to get out of that with parameterizations that we currently know enough to apply.
So then how do you use your models to help manage the forest?
I think that it's very much stand specific. I think that if you're concerned about strawberry guava in particular coming up, and there is some strawberry guava that's in those plots, then you're going to need to incorporate plot data that has strawberry guava in particular. And you can apply this method to plots that have that demographic information. I think that for the lob-away plots, the interest is in managing invasives so we can see what natural dynamics are. And so in that case, the management would really be on the questions would really be on how do we manage the native forest with the introduction of this new pathogen? How does this new pathogen reality feed into natural dynamics? If that makes sense.
Thank you, Brandon. There is a question from Gina in the chat. She's asking, do you think that there are social or cultural influences on these forests? For example, access by hunters or non-timber species gatherers?
Yes, I do. The non-timber species gatherers, it would be interesting to look at that in particular. It'd be interesting to run an experiment looking at selective logging, for example. I know you said non-timber species, but looking at selective logging of things like koa or something, and see if that has any real impact on dynamics or what thresholds might be for low-level harvesting. The social and cultural influence, I think, definitely there's a lot of work that's coming out looking at the relationship between fencing and pig hunting and the spread of rod. And that's not my work. I can't comment on it too much, but I think there's a lot of cultural implications and social implications there with respect to managing, especially the mature ʻŌhiʻa forest.
Mahalo. Okay, one more from Rebecca Ostertag. Hi, Brandon. She says, nice talks and thanks for sharing. What do you think are some of the data limitations with Hawaii plots of these types?
The biggest limitations are on because these, thanks for the question, Becky. The biggest limitations have to do with things that aren't measured in these sorts of censuses. And I think that the strawberry guava was definitely that's a limitation, especially really herbaceous or smaller, like shrubby things that come in and take over. There's some of that that's being measured, but I think it's hard with these methods to capture those really intense like invasion and takeover events. And second, looking at recruitment, real recruitment happens at the seedling stage, which I think is very important. But because of the cutoff, there's a cutoff of 1 centimeter DBH before things are included in the census. And so recruitment from the perspective of this data set is really recruitment into like the seedling stage. And so it's not, we're not really seeing the dynamics that happen at any size below that. We just see the emergent consequence of seedling dynamics and what their effect is on the next size class.
Mahalo. There's also a link from Stephanie. She is giving some additional information about strawberry guava being released, some previous work from the koa looper moth defoliation event. So if anyone wants to check that out, she has linked in an article there. And then Megan is asking, she'd like to revisit the rod statistics concerning the Laupahoehoe plot. Do you see a baseline percentage of dieback per year, or are numbers accelerating over time?
I do not know enough to answer that. I know that there's a ton of rod work and there's tons of rod plots. So I do think that people know those numbers. I think that Flint Hughes would have those numbers. There are some, I don't know if they're in Loboy, but they're in the Loboy Forest. And so, yeah, those numbers exist, but I don't know them. Sorry.
Yeah, and just to add to that, we did have Flint Hughes on last month. So if you folks are interested in listening to what he had to say, his presentation is on our HETF YouTube of all of our recorded webinars. And it looks like Liat, I'm hoping I'm saying that correct, has their hand raised. So let me unmute you.
Hi, thank you. And yes, that was a correct pronunciation of my name. Thanks, Brandon, for your talk. I just wanted to clarify, I wanted to make sure I was understanding your response to Julie's question a little while back about the management implications of the data that you presented to us today. So what I got from that is the way that you're looking at the data supports like a theoretical baseline in forest dynamics, like without the effect of invasion.
Yeah, I think that it can be used to answer questions with invasion. I just don't think strawberry guava is a very good question because the stems are so small and because it takes over the way it does other things like there are some tropical ash on the plots, and so I don't know if those are being actively managed, but if they're not, then we can look at the recruitment and invasion of tropical ash in that plot and how that connects to dynamics of ʻŌhiʻa and koa and like overstory species. I'm sure if there were other species on the plots like Albizia, you could use it, those sorts of connections. There's a limitation with the data in terms of like the size of things though. And so I think specifically for the strawberry guava, that would be difficult to look at. Does that make sense?
Yes, thank you for clarifying.
Mahalo everyone for your questions. So if there are no more, I guess we will end a little early. And, I just want to thank you folks all for attending this webinar. We will be uploading the video later to the YouTube channel that we have. And if you folks want to connect with Brandon for further information, I think we would be comfortable sharing his email so you can reach out to us at the HETF. And thank you again, Brandon, for taking the time to share with us.
No problem. Put my email in the chat there.
Thank you. And then one last thing, I will share the link to our Google form one more time, just in case people didn't catch it at the beginning of this. And so we do ask for feedback about our webinars, really any comments or, additional questions or requests are more than welcome. so that we can incorporate this and create better resources for everyone. Thank you all so much for your time. Take care. Aloha.